Hi, welcome to another episode of Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Now today, what we're going to talk about is how do cells communicate long distances, right? In the last video, we talked about how they did it over short distances through paracrine signaling, but today we're going to talk more about the endocrine signaling. And how do they do this? Well, they release hormones, right? And you might be familiar with some of these hormones, including insulin. Insulin's used whenever you could drink a Mountain Dew. You get all that sugar in your blood, right? Well, what happens is when that sugar gets absorbed in your blood, your pancreas releases insulin, and that causes the cells of your muscles and your fats to pull in the glucose out of your blood. Another one is thyroxine. It helps regulate your metabolism. So if your thyroxine levels go too low, your metabolism might slow down and you might gain weight. Thyroxine levels get too high, it's the opposite effect. You've probably heard of estrogen, testosterone, serotonin. How about adrenaline? Man, yesterday my adrenaline was flowing. I fell through. I'm not gonna say what I fell through, but I fell and ended up having to go to the doctor to get fixed. And yeah, my adrenaline was flowing pretty good right after my fall. One of my favorite hormones is actually not a lipid. It's a protein, or at least it's a peptide hormone. It's only 9 amino acids long, and it's called oxytocin. And oxytocin is often called the love hormone. So when you look deep into your partner's eyes or your dog's eyes, it'll work too, and you get all those warm fuzzies, or your children's eyes, that's from oxytocin. But oxytocin is also involved with not just social bonding, but reproduction and childbirth. So oxytocin is one of the hormones needed to cause contractions. So a few weeks ago in October, our second child was born, and we had to induce labor. And the way that they induce labor, they gave my wife Pitocin, which you may have heard of. Pitocin is just a brand name for oxytocin. So I would see them ramp up her oxytocin levels, and that caused her to go into labor. Wow, amazing, isn't it? Okay, these are examples of several different types of hormones. Now, hormones are a type of signaling molecule, and a general type of signaling molecule is also called a ligand. So whenever you hear the word ligand, think signaling molecule. Now, we've got this endocrine signaling between these distant cells, right? So you've got your adrenal gland in your kidneys. They'll release adrenaline, and that will affect cells throughout your entire body, from your lungs to your heart, all over the place, right? So these hormones are transmitted through our circulatory system, right? And this type of long distance signaling is not the fastest one. Of course, the fastest one is our nervous system, but these might have a bit slower response, but they are crucial for multicellular life. Now, what I'm going to focus on today is not so much of the different types of hormones or the tissues or glands that produce them. I'm going to talk more about how do cells receive these signals and how does that elicit a response. So the first thing you have to have is signal reception. The cells have to have some way of receiving the signal. Then you've got signal processing. This is transduction. You got to get the signal into the cell and amplify it. And then you have some type of response. Why send a signal if you're not going to have a response? And then signal deactivation. And so this lecture is going to focus on these. So let's start with signal reception. Okay, to receive a signal, you got to have a receptor that binds to the hormone. Now think about this. There are radio waves all through the air right now. I mean, all your favorite radio stations, all your TV stations, cell phone, I think they use microwaves, but there's all this information actually passing through you right now. Are you listening to the radio inside your head? Oh, I don't know. Maybe you're thinking of a song. But the point here is we have no way as a biological system of intercepting those radio waves and interpreting them. But we have equipment that can. That can pick up those radio waves. Our cells are the same way. Our bodies are the same way, because we have all kinds of signals that are constantly moving throughout our body. There are lots of these hormones in constant flow. And for a cell to respond to them, it has to have the right type of signal receptor, which is often a protein. And a lot of these are lipid soluble, and some are lipid insoluble. So a lipid soluble hormone, they can actually cross a cell membrane, all right? So the receptor for those will be inside the cytoplasm. Now, nicely, it can get through the cell membrane very quickly, like a steroid hormone like testosterone or estrogen, and they will get in there and they can quickly respond, and there's all types of responses these can cause. Now, the next one, of course, is going to be the lipid insoluble signals. Lipid insoluble, it just means it won't dissolve in a lipid. These are hydrophilic molecules, right? They can dissolve in water, and that makes sense, right? Because when you think about our circulatory system, what is it mostly made of? Water. So if you're going to send a signal, you want it to easily travel through the water, right? You don't need another carrier protein for these things. So a lipid insoluble ligand or signaling molecule, also hydrophilic, it cannot cross cellular membranes. So instead, the cell must have a receptor on the surface of that membrane to receive a very specific ligand. Now, these receptors are dynamic, okay? And so they can respond to the environment. So here's an example. Part of the problem in our society is we have poor diets, often way too much sugar. And as you know, when you drink sugar, glucose is quickly absorbed into your blood, and that causes your pancreas to release insulin. And then insulin triggers a pathway or triggers a transport molecule to bring glucose into your cells and lower blood sugar levels. However, your cells can become insulin resistant. So the number of receptors for insulin on your cells can decline over time if you're constantly drinking too much sugar, and that leads to insulin resistance. So this can happen for a lot of different types of protein receptors, right? They can decline in number over time if there's a constant signal all the time. The other thing that's important too is that these are proteins and genes code for proteins. So you can imagine that a defective gene could lead to a defective receptor. Now, also, you'll learn this in genetics, but the amount of the product that's made, in this case of your receptors, is also controlled by other genes. And if those regulatory genes become defective, you can have too few receptors, as in the case of hypercholesterolemia. Well, hypercholesterolemia, that's a long one. It means you have too much cholesterol because your cells do not produce the receptors for LDL, which is your low density lipoprotein, to bring in cholesterol. So you build up cholesterol, you build up plaque, you get heart disease very early, and you die in your 40s if you have a mild form of the disease. Another example of receptors is cancer. We'll talk about this in the HER2 gene or the HER2 gene, which causes too many of these RTKs, something we're going to learn about, that causes these cells to have a very strong growth signal. And another thing, receptors can be blocked. Like a beta blocker. Somebody that's on a beta blocker, they're doing it for blood pressure. My dad, he's on a beta blocker. He's had heart damage over his lifetime, and the doctors do not want his heart to ramp up too high because it would stress it, could cause another heart incident. So his beta blocker blocks adrenaline. So if he gets excited, his heart rate doesn't go up. It's pretty wild. And we also use some types of signal transduction blockers as well to help treat cancer, to slow the cancer cells from dividing, to keep them from going so fast. In signal transduction, we've got two major types. We've got the G protein coupled receptor and a receptor tyrosine kinase, okay? And what they do is they receive a signal and then transmit it through the membrane. Let's talk about some of these signal transduction molecules. They're a fascinating class of molecules. Did you know that there are 800 known G protein coupled receptors? Now, the G protein coupled receptor is a type of transmembrane protein. It's also an integral membrane protein because it's permanently attached to the cell membrane. Now, the G protein, that is a peripheral protein because it's on the cytoplasm and it can actually move around, but it's associated with the G protein coupled receptor, right? So we got these two things here together. And what they do is when a specific ligand, specific chemical signal binds to the G protein coupled receptor, GPCR. We can call it GPCR, right? And when it binds to it, what it does is it causes a conformational change. And that conformational change is transmitted through the membrane, okay? And then once it gets there, it can activate the G protein. Now, one way that we often activate and deactivate proteins is through a phosphorylation event. Adding a phosphate group usually activates a protein and turns it off. And when we remove the phosphate group, it deactivates that particular protein. Now, in the case of the G protein, it's still activated what's called guanine triphosphate, GTP. That's the active form. But instead of phosphorylating GDP to GTP, when the G protein coupled receptor is activated, when it does its shape change, it cleaves off the GDP and puts on GTP. So make sure you got that down. It's not phosphorylating GDP to GTP. It is cleaving off the GDP and putting on a GTP. And at that point, the G protein coupled receptor becomes active. Now, remember, the G protein is a separate protein in the cytoplasm. And the reason why the G protein coupled receptor gets its name is because its activity is coupled, right, to that G protein. And the G protein is a type of peripheral protein. It is not an integral protein. So it's not permanently attached to the cytoplasm. It's not permanently attached to the cell membrane. Now, once the G protein is activated, guess what? It's going to go on and basically interact with a second enzyme. And what it does once it starts activating that second enzyme, it activates it and sets off secondary messengers. And then the G protein, it becomes inactive when GTP loses a phosphate group and goes back to being GDP. Now, remember, in this case, it's losing this third phosphate group. It's not having it removed. And the reason why that's important is because those phosphate groups, remember, it's a phosphorus surrounded by oxygens. These things are negatively charged. And when you stick these negatively charged molecules on another molecule, if it's got negative charges, they'll repel each other, and that will cause a conformational change. It'll cause a shape change. That's why these molecules have two positions, an on and an off, and you turn them on and off by the position of these phosphate groups. Now, what's interesting here is once our secondary enzyme is activated, it sets off a series of events by activating what's called second messengers. Now, a second messenger, they're called second because they occur inside the cell after the first messenger, which is the ligand that activated the G-coupled protein receptor, right? So the second messengers are second. They occur inside the cytoplasm of the cell, and there are various types of these second messengers. Cyclic AMP, and we'll talk about some of these in later chapters. ITP and calcium ions. Calcium ions are actually a very important second messenger, and in fact, they're used in muscle contractions. They're held inside your sarcoplasmic reticulum. You may not have heard that one, but that's part of a smooth endoplasmic reticulum that holds calcium ions. And when they're released into the cytoplasm of a muscle cell, well, they cause muscle contractions. Okay, so a secondary messenger can serve multiple roles inside of the cell. And then, so that's how a G protein coupled receptor basically works. It takes a ligand from the outside of the cell, binds to the G protein coupled receptor. It causes a conformational change, which then activates the G protein. The G protein then interacts with another enzyme that sets off or activates the second messengers inside the cell. So understanding how G protein coupled receptors work, here's a couple of examples of why it's important. People get asthma attacks. One way we can treat asthma is by using a blocker that blocks to a specific G protein coupled receptor, and that will allow the muscles to relax, the smooth muscles to relax. And instead of having your lungs contract and all your things contract and constrict, it can cause them to relax. And then interestingly, it's weird, but there is a group of people, there's a population of people, mostly descendant of Europeans, that are resistant to HIV. You guys, it's not so much on the forefront anymore. I'm a child of the 80s. I was in high school in the 80s. It was a big deal. And because if you got AIDS, you died. And it turns out that some people are immune to AIDS. And the reason why viruses are very specific in terms of which cells they usually attack. And in this case, it attacks, of course, your helper T cells, which takes out your entire acquired immune system. But the way they do this is by attaching to a very specific G protein coupled receptor on the surface of the cell. Well, it turns out that there was a really bad plague that killed like 1 out of 4 people in Europe a few hundred years ago. The Black Plague, the Black Death, the bubonic plague. Well, at any rate, some people that survived that had a mutated form of this G protein coupled receptor. And those descendants that inherited that mutated form can't get AIDS. The HIV virus can infect them, but it cannot enter the helper T cells because it can't recognize a specific protein. So even if you get the virus in you, nothing happens if you had that particular variant. Pretty interesting stuff. Okay, let's move on to the next major class of signal receptors, and these are the RTKs. RTK, what does that stand for? How about receptor tyrosine kinase? Okay, I know RTK, receptor tyrosine kinase, there's a lot going on here. This is another type of integral transmembrane protein. It's permanently attached to the membrane. It spans the membrane, so it's a transmembrane protein. And it's also called a kinase. Now, kinase is a class of enzymes. There are perhaps thousands of kinases. And kinases, there's thousands of different types, but they have one basic function. They add a phosphate group to a molecule, right? They add phosphates. Now, the reason why there's so many different types of kinases are lots of different molecules are activated or deactivated using phosphate groups, like a sodium-potassium pump. There is a specific kinase that attaches phosphate groups to a sodium-potassium pump. Well, the receptor tyrosine kinase, it's a receptor, so it's receiving a signal. Tyrosine is one of the 20 amino acids used by all life, kinase, adding phosphates. So RTKs, receptor tyrosine kinases, I'm just going to go with RTKs from now on. RTKs are also an enzyme because when a signaling molecule, a ligand, binds to it, it causes a conformational change. It's actually a dimer. There's these two parts of it. And when the signaling molecule comes together, the dimer comes together like this. And when it comes together like this, it's activated. And part of its activation process, it phosphorylates the tyrosines. Receptor tyrosine kinase, right? Because we're phosphorylating the tyrosines from a signaling molecule. That's where it gets its name from. These signal receivers are very important, especially for something called a mitogen. Mitogen, these are like growth factors, mitosis, that cause cells to grow and divide. So as you can imagine, they're very important for receiving growth signals. This, of course, is a different view of a RTK. You can see that it's existing as two different parts. And then when the signal molecule comes in, it's going to cause a conformational change. And on the right, that is the amino acid, tyrosine. Look for the carboxyl group, look for the amine group, and then you can see that it's got a ring structure as its side chain. Now, once the signaling molecules come in, you can see that the dimer comes together, that it comes together and forms the dimer. In this case, it becomes activated, and it then phosphorylates those tyrosine residues, and this is now activated. And once this happens, once RTK becomes phosphorylated, it's now active, it can set off a series of responses within the cell. And in some cases, it can even activate G proteins. But like I said, one of the things it does, it sets off different types of reactions within a cell. So you might get multiple responses. And specifically, it can set off something called a phosphorylation cascade. This is also a signal amplification as well. Now think about this. You might not have much of a signal. There just might not be much coming in. So you've got very few receptors on your cell, and one of the two receptors receives a signal, and it becomes activated and then sets off this phosphorylation cascade. What that means is I've got one molecule is going to become activated. It's going to in turn activate another molecule by phosphorylating it. It's going to in turn turn on another molecule by phosphorylating it, right? This is our phosphorylation cascade, as all of these molecules become activated through a phosphorylation event. And it's also a signal amplification. If I have one molecule gets phosphorylated, it in turn phosphorylates and activates 10 additional molecules. And those 10 additional molecules each activate 10 additional molecules. Now I've gone from 1 to 10 to 100 to 1,000. You see where I'm going with this. The signal can be amplified within a cell quite rapidly. And this type of phosphorylation cascade is also very important for cell division in terms of getting the events going to get the cell to like go through the cell cycle, which is what we're going to learn in the next few chapters. Now, this is why this is important. Let's make some connections here. You could have two women get breast cancer, and they're both at the same stage, both similar types, same age women, same health. One dies in a year, and one they remove it and lives the rest of her life with no problem. Why is that? Why do some forms of cancer grow much more aggressively than others? Why do some have good prognoses, others don't, even though we call them breast cancer? Well, it turns out that cancer is a generic term for over 100 different diseases, and even something like breast cancer that occurs in the breast tissue, could have several different causes. One reason for aggressively growing breast cancer is a variant in the HER2 gene that causes an overproduction of RTKs. Now remember, RTKs, what we just talked about, they receive growth signals. So in aggressively growing cancer cells, they have too many RTKs, and the cell has this really strong response to grow and divide. And that makes these cancer treatments difficult to treat. Let's move on. So signal response, let's just recap this really quick. You've got these signals inside your body. They're floating around. To receive that signal, you have to have a receptor. And if the signal is hydrophilic or lipid insoluble, those receptors are on the surface of the cell. They bind to that signal, and then they transmit it into the inside of the cell, where they will set off secondary messengers. And this is transduction through the cytoplasm until you get a cellular response. And cellular responses could be incredibly varied. Testosterone might tell you to build more protein. It could be a mitogen and a growth factor. Enter into mitosis. So there's lots of different things that can happen here. And even plants. Plants are multicellular organisms as well, and they also send out signals throughout the organism. And one of them is plants, they have to have a constant supply of water from their roots up to their leaves. And the water exits the leaves. But out here we get dry, and these roots get drought stressed. So the roots will actually send a signal, it's called abscisic acid, up to the leaves. And then these tiny openings on the other side of the leaf are called the stomata. And the stomata is surrounded by a guard cell. And when the guard cells receive that chemical signal from abscisic acid, it'll cause them to close, and the plant can conserve water. And we learned about C4 plants. They can close that stomata, and they can continue doing photosynthesis through C4 pathways, drawing carbon dioxide down much lower. And then lastly now, we have what is called signal deactivation. And that makes sense, right? Because you don't want your cell signal constantly on. It's got your response can't stay on forever. So you have to have some way of deactivating it. Now with the G protein coupled receptors and these G proteins, once they interact with that secondary enzyme to set off the second messengers, to activate the second messengers, that G protein is then inactivated with the loss of a phosphate group on the GTP. Now remember, GTP is guanosine triphosphate, ATP is adenine triphosphate. Remember, these are both purines, so they're really, really similar. Okay, and then earlier I talked about the phosphorylation cascade and how you have these kinases that are adding on phosphate groups to molecules to activate them. Well, one way that you can deactivate a molecule is by removing the phosphate group. And there's a whole class of enzymes called phosphatases. There's that ASE again, right? You're starting to see the kinase, phosphatase, ATP synthase. ACE means it's an enzyme. Phosphatase removes phosphate groups from activated molecules and in turn deactivates them or turns them off. And then lastly, there's some crosstalk going on with these cells, right? Now here's one way that is different from your radio. Your radio dials into one radio station, right? You're going to listen to like Gulf 104 if you're in Florida, or you're going to listen to 104.1 The Edge if you're out here in New Mexico. Oh yeah, New Mexico, you got way better radio stations than the rest of the country. Our radio stations out here are amazing. But the point is, you tune into one station. If you're watching TV, you're tuning into like one station. I don't know what you guys watch. Um, I don't watch much TV. I tile into Netflix. Now, cells are different. Cells don't receive just a single signal at one time. They often receive multiple signals all the time. And this can cause many responses, and these responses start interacting with each other. And so, for example, you might get a signal that says grow and divide. You might get another signal that says, no, stop that. So one pathway can inhibit or limit another pathway. And you can have just the opposite as well. So you've got one pathway stopping another one, or you've got another pathway amplifying the other pathway. Or one pathway amplifying a second pathway. So the point that I'm trying to make here is that cell signaling is very complicated. There's lots of different signals. There's over 800 G protein coupled receptors alone that we know about, and that cells receive multiple signals and have lots of crosstalk. And so these cells are responding to multiple stimuli at once. Okay, now that's been a very brief introduction into cell signaling with a focus on the receptor molecules, the G protein coupled receptor and the receptor tyrosine kinase. And then in my next lecture, I'm going to talk about apoptosis. All right, until next time, this has been another episode of Tom Kennedy Science.